You've probably heard the expression genetic code. Um, whenever I heard the expression genetic code, sometimes to me it sounds like they're speaking poetically. But really, the term genetic code is quite literally what we see in DNA. And here's why. We, like I was there, life created this system so that there would be what is known as perfect fidelity. Okay. Perfect fidelity means no mistakes when you are copying the DNA. That was really, really important because if you make a mistake in the instructions to how to build a protein, imagine if every time IKEA made a copy of their instructions, there was a little mistake in them. After 5 different copies, 10 different copies, no one could put anything together. So since replication requires life to make copies, we needed a system that that could be perfect. So in order to do that, we only have 4 different nucleotides. But the problem is we've got 20 different amino acids. So in order to use this system that has 4 different unique nucleotides to give us the information about 20 different amino acids, we had to assign them three, three letters at a time. 3 nucleotides in a row in a coding region of DNA or in a coding region of RNA is known as a codon. 3 different nucleotides in a row that are a part of a coding region are known as a codon. Little tiny code word, okay? So every little 3 letters, like what we did Gattaca. So GAT, that's a little code word that means one of the amino acids. And it had to be 3 in a row because we've got 20 amino acids. So I'm not going to go through the math with you, but if you were to use one nucleotide to every amino acid, you got 4 nucleotides, so you could do 4 amino acids. That's not enough. If you could do 2 nucleotides for every amino acid, then it would be, you could do 8. You could do 8 amino acids. Don't write that down, okay? But if you do 3, you actually have 64 unique little code words that you can make. So when it comes to using them 3 at a time, you can end up making 64 unique little 3-letter words like GAT, GTT, GGT, right? You can make 64 of them. So life has created a code, and that code will say something like, for example, if the DNA says TAC, that's code for put a methionine here. Every amino acid has got at least one codon that identifies it. So here we go. You might recognize this because it's in the lecture part of the class. This is a protein that I discovered. I know, it's pretty exciting. Anyway, these letters here that are pretty far apart, each one of those is a single letter abbreviation for an amino acid. So you don't have to worry about the single letter codons for amino acids, but each one of them is one of the amino acids. And what you'll see above it is the DNA, the coding sequence of the DNA for the protein that I discovered. So this is found in your genome. So ATG, I told you, that stands for methionine. So every time we look in here and we see the letter M, we will see ATG right above it. So you see, this is a code. So all of this stuff that's all in Gattaca, right? GAs, T's, and C's, that's all in code. It's in the code of nucleotides. And underneath it, we have got the decoded protein. And this is the primary structure the amino acid sequence of the protein that I discovered. See, there I am. Okay. All righty. So that leads us to what is DNA's second big job. Its first big job is just to copy itself perfectly whenever the cell divides. But its second job is to be the source of information for the ribosomes to get the instructions for how to build a protein. Now, keep in mind that even here, seems like a whole lot of DNA. This is nothing. This is 700 bases, and keep in mind that even one of your chromosome is millions of bases big, okay? So but the ribosome's job is to build a protein. And all of this stuff right there, that's the information that the ribosome's going to need. And that leads us to the second job that DNA has. The second job that DNA has is to give instructions to the ribosomes. The nucleus will decide, I want this protein made, or I want that protein made. Let's imagine that your nucleus is like the Cerritos College Library. And the way the cell works is periodically, the library just sends out, like a big paper airplane, the instructions for children build this. And we're all the ribosomes, and whoever catches that airplane, it's our job to build that. Making that paper airplane, making that little copy of just one chapter of one of the books in the library, just the instructions for building one protein, that is called transcription. That's called transcription. I can't, okay? All right. So DNA versus RNA. We haven't talked about RNA yet, except for it's a nucleic acid. The thing is, DNA is so valuable, it needs to stay inside the nucleus where it's safe. So here's our nucleus head honcho, and she is holding the entire set of plans to make an entire you. Now, here comes Mr. Ribosome. Mr. Ribosome's job today is going to be he's just going to build a wall. He does not need all of the blueprints for making the entire 20-story building. He just needs to know, boss, where do I put the wall today? Right? The answers to what this little ribosome needs to do, the answers are inside of the DNA. And so the DNA is going to say, I am just going to send out just the instructions that are in one gene, okay? He just needs to know where to build the wall. So I'm just going to make a copy of that part, that little single part of all of those instructions, it's supposed to move. It didn't move. And I am going to send it out to him. And the way it gets sent out in the form of RNA that is supposed to move, okay? So we will do it manually. So the nucleus is going to take all of that DNA, and it's going to find one little segment, and it's going to say, okay, this is the part he needs. I'm going to make that little part, and I'm going to send it out to him, and he is going to just have that little part. Now, DNA is made out of DNA nucleotides so that it is permanent. RNA, those instructions we send out to him, we want those instructions to be temporary. We want him to read them, to build the wall, and then throw it out. Because tomorrow he's doing a different job, okay? So each gene is the instructions to make a protein. The ribosome needs the instructions to make a protein. The ribosomes are not in the nucleus. And the nucleus is not going to trust this fellow with all of the DNA. So the nucleus is just going to make a little tiny disposable copy of one gene, and they're going to make it in the form of RNA and send it out to the ribosome. And yes, ribosomes can be free in the cytoplasm, or they can be attached to the endoplasmic reticulum. All right? RNA. They're very similar. Both DNA and RNA are made out of nucleotides. DNA, phosphate sugar base, but the sugar is deoxyribose. RNA, phosphate sugar base, but RNA, the sugar is ribose. And the little R at the beginning of RNA stands for ribose. Okay? DNA is double-stranded. RNA is single-stranded. DNA uses these 4 bases, Gattaca. RNA is close. DNA uses G, A, T, and C. RNA uses G, A, U, and C. All right? So with DNA, the binding rules with DNA, if I had another piece of DNA, it would be that's not right. Let's write it in a better place. Let's say we have G, A, T, and C. That's going to be our DNA. The complementary strand of DNA would be C, T, A, and G, right? Because wherever there's a G, there's going to be a C. Wherever there's an A, there's going to be a T. But for RNA, the complementary would be C, U, A, and G. Because the C and the G still binds together, but now there is no T, so the A goes with the U. Right? Now, if I ever ask you, is there ribose in DNA? No, there's not ribose. Now, it's true the letters ribose are in the name of the sugar deoxyribose, but there is no ribose there. This is ribose. Ribose is a sugar that's got an extra oxygen there. That sugar ribose is not found in DNA. It's only in RNA. This sugar's name is deoxyribose. See, it's missing an oxygen. It's like, if you really love Justin Bieber, don't make a mistake and go into the I hate Justin Bieber fan club, okay? Because you'll be in the wrong place. They both have Justin Bieber in the name. They both have ribose in their name. But ribose is ribose, deoxyribose is deoxyribose. There are 3 different kinds of RNA that are important to our story. Ribosomes are made out of this kind of RNA. Do you see that little R at the beginning, the lowercase r at the beginning of that RNA? So rRNA. That lowercase r stands for I'm the one that makes up the ribose. And ribosomes are made out of ribosomal RNA, rRNA, and protein. That little copy that I was showing you here, that little copy that the nucleus sends out, that is messenger RNA. That is the message from the nucleus. That is the nucleus saying, build this for me, peasants, okay? mRNA, messenger RNA, those are the instructions. Transfer RNA is a special little robot kind of RNA whose job it is to escort amino acids one at a time to the ribosome so the ribosome can build the protein. All righty? We're going to start there at the beginning of the next video.